Today we are going to talk about basic electrophoretic techniques to separate proteins and study proteins. So, what is electrophoresis? It is one of the powerful technique for protein separation, and the separated proteins can be visualized after subsequent staining steps. It is based on the principle of migration of the charged proteins in the electrical field. As we have studied in the last two lectures some basics of amino acids and proteins, and then we moved on to study the proteins and proteomics. You have seen that now there is emphasis on looking at thousands of proteins simultaneously. That is very relevant, especially if you are looking at the complex biological problems, because the proteins work in the interaction, they work as a part of signaling pathways. So, studying proteins together in totality becomes very crucial. So, there are many techniques which are evolving which aims to study proteins in very high throughput manner, and they try to utilize protein properties to separate them well. One of such technique is known as two-dimensional gel electrophoresis or 2DE. 2DE is a powerful electrophoretic separation technique that separates proteins in two directions. The isoelectric focusing, which carries out in the first dimension and separate proteins on the basis of their unique isoelectric points, and then the SDS page, which separates protein on the basis of their molecular weight, and that is known as the second dimension separation. So, because we are utilizing two different properties of proteins, in the first dimension isoelectric point and in the second dimension the SDS page, this technique is known as two dimensional electrophoresis. So, if you want to do an experiment based on two dimensional electrophoresis, this kind of workflow can be followed. You have extracted the protein, you have done all kind of quality control checks to ensure that proteins does not have any contamination, like you do not have nucleic acid contamination, carbohydrate or lipids as a part of the protein, and the protein does not have even salt, which may even interfere in your assays. Then you have to quantify the protein, and once you know that now you got a decent clean protein with good concentration, that protein is now ready for the further complex analysis. And now your intention is to separate those thousands of proteins present in that mixture using two dimensional electrophoresis. So, to do that, the very first step you will do isoelectric focusing in the first dimension. Then you want to prepare the strips to separate them in the SDS page, and to do that you are doing an equilibration of IPG strips. Then the next step comes for the second dimension separation using SDS page. Then you want to visualize the gels using staining techniques, and then you want to do the image analysis using various software, and then those protein which looks interesting to you, you want to excise those and then identify them using mass spectrometry. So, this kind of a typical workflow. So, very first thing that you want to look into which kind of immobilized pH gradient strips you can use for doing the protein separation. These are the acrylamide based plastic strips on which the different pH buffers are coated, and then they comes in different pH range. For example, it can be pH 3 to 10, it can be even 4 to 7 physiological range, and they also come in different length. For example, you can always start with a smaller length of 7 centimeter, but for the actual experiment you can choose the large length of let us say 24 centimeter, and then your intention is to resolve the proteins on the large gels, so that you have good separation of various protein spots. So, these immobilized pH gradient strips, they are very stable, they are much more durable, and because the gel is prepared with a plastic backbone, that actually ensures that the pH gradient is fixed in the place. It provides high resolution, and it has also improved lot of reproducibility in different laboratory comparison, which was one of the drawbacks in the earlier 2D technologies which were based on the tube gels. These IPG strips provide you good capacity for loading lot of proteins, and therefore, now you are ready to separate proteins in the first dimension. But before you want to when you are doing an experiment, the very first step you want to do, you want to take your protein in the solution form and you want to get it immobilized in the IPG strip or immobilized pH gradient strips. To do that, your very first step is rehydration step. So, you want to rehydrate the protein solution onto IPG strip in the overnight in a reswelling tray, and that you can do either using a passive rehydration method, or you can do using an active rehydration method. So, in passive rehydration there is no voltage is applied, whereas in case of active rehydration you can apply the low voltage. Then after you have put your protein solution, you have added now the IPG strip, and then you have covered it with some mineral oils, so that the proteins are not solidifying on the edges and it is not getting evaporated. So, then overnight you are leaving it, so that proteins slowly get absorbed in the IPG strip, and then the next day when you want to start your first dimension separation, then you are ready to do the isoelectric focusing. Let us also see some of these details of doing rehydration and IF in the animation form.
Prior to isoelectric focusing in 2DE, the commercially available IPG strips must be rehydrated. This can be done either by passive rehydration or active rehydration. In passive rehydration, the IPG strip is placed with its gel side downwards in a well containing the protein sample, reconstituted with a suitable buffer solution. This is then covered with mineral oil to prevent the gel from drying up and left overnight for 10 to 20 hours depending on the length of the strip. In active rehydration, the protein sample is added to the strip via a sample cup, followed by cover fluid to prevent the gel from drying up. This is then placed in the isoelectric focusing instrument and low voltage is applied, which allows the strip to take up the protein sample. Active rehydration is also performed for 10 to 20 hours depending upon the length of IPG strip being used. These loaded strips are then focused on an isoelectric focusing unit by passing current. The various proteins of the sample mixture migrate in the electric field and comes to rest when the pH is equal to their pI, that is, they become neutral and are no longer affected by the electric field. Progress of electrophoresis is monitored by means of a tracking dye like bromophenol blue BPB, which is a small molecule and therefore migrates ahead of all other proteins.
So, let us talk about isoelectric focusing. So, as we have studied in one of the earlier lectures that these amino acids have different side chains, provides the positive and negative charge. So, proteins have overall net negative or positive charges, and the pH determines the ionization states of these amino acids and therefore, the charge on the protein. So, isoelectric point is the pH at which the net charge on the protein is 0. So, when pH equals to pi, there is no mobility of these proteins, and isoelectric focusing or the IEF uses this particular fundamental property to separate the proteins, and depending on what kind of pH strip you are using, you can separate either in the pH range of 3 to 10 or 4 to 7. So, when you are running proteins on SDS page gel, when you want to separate only based on the molecular weight, you want to boil the protein, you want to denature the protein, you want to separate the denatured protein on SDS page gel. However, in case of 2-dimensional electrophoresis, because you have started on the IPG strip, so you cannot boil them. So, you have to prepare the IPG strip before you load them onto the SDS page gel, and that is done in a process known as equilibration. So, this is one of the conditioning step which you apply so that the proteins which are separated by the IEF, they are now prepared to be separated in the second dimension based on the molecular weight. So, the objectives of equilibrating these proteins onto the IPG strip are to coat the proteins with SDS for the separation which you use for the molecular weight basis. Then you want to also cleave the disulfide bonds, which is inter or intra chain disulfide bonds, and you want to also alkylate the sulfhydryl groups of the cysteine residues. So, let us look at some of the recipe which is used for doing the equilibration. The first equilibration step involves addition of DTT, and that DTT along with you are adding the SDS, you are adding the tris HCl and glycerol, and in the second dimension you are adding the iodoestamide. This iodoestamide is going to prevent the protein reoxidation. So, whatever you have denatured and those bonds should not be reformed again, and these alkylates the residual DTT to minimize the vertical streaking as well. So, you have to perform both first and the second equilibration steps, and ideally you can do it a repeat process of that to ensure that your reduction and alkylation has happened properly. So, once the IPG strip, you have already focused the protein based on the first dimension property, and now you have also prepared the strip to be separated further based on the molecular weight. So, now you can use the second dimension protein separation property, and that itself is a very interesting technology which is known as SDSPH, SDS polyacrylamide gel electrophoresis. So, the proteins when they exhibit different molecular weights depending on the amino acid composition, this property can be utilized to separate the proteins using SDSPH gel. And this electrophoretic method, which is SDSPH, is aiming to separate the proteins based on their molecular weight. So, now whatever we have separate proteins based on the PI values, they are going to get further separated based on the molecular weight. So, SDSPH is one of the widely used electrophoretic technique. It separates proteins according to their size. The detergent SDS, sodium lauryl sulphate, is negatively charged. In case of if you are just directly separating proteins on the SDSPH gel, you can simply boil the protein in SDS and beta mercaptrethanol. In case of 2D electrophoresis, you are going to prepare the IPG strip with the equilibration steps. After reduction and alkylation steps, now those IPG strips are ready to be separated further on the second dimension SDSPH. Just kind of brief about SDSPH, some of the chemicals which you use to make the gels. What is the role of each of these components? For example, acrylamide, it is providing the matrix. Bisacrylamide is a cross linking agent. Ammonium persulfate or APS, it initiates the polymerization process. The SDS is a negatively charged detergent which makes the protein rod shape and negatively charged, and then beta mercaptrethanol, it breaks the disulfide bonds. So, some of these chemicals are used for the SDSPH process. I think it is a good idea for you to know the role of them and how they work while performing the SDSPH gel. So, you have started with your protein immobilized using in the rehydration process onto IPG strip. You have done the first dimension separation using isoelectric point, equilibrated the strips to prepare them, and now you have separated proteins in the SDSPH gel based on their molecular weight properties. So far everything whatever you are doing, it is all on the transparent gel. You have literally no idea that what can be seen on those gels and what you have separated, until unless you are adding some staining reagents which can visualize the gels. So, therefore, different type of staining reagents becomes very important. The most commonly used staining reagent is Comassi brilliant blue, but you can also use silver staining, you can use different sensitive dyes like cypro rubies, and even if you are looking at some modification at the PTM level, you can use even ProQ diamond, or you can look at for very sensitive detection, you can use even cyanine dyes. Let us watch the following animation to understand this concept better.
The IPG strip is equilibrated in a reducing agent like DTT, followed by an alkylating agent, iodoacetamide, which prevents reformation of the reduced bonds. This strip containing the separated proteins is then placed on the SDS polyacrylamide gel slab and subjected to SDSPH by applying a direct current between 100 to 350 volts depending upon the size of the gel. Any proteins that may have been present as a single band on the IPG strip due to similar isoelectric points can now be separated on the basis of their molecular weight, with smaller proteins migrating farthest. View of a sample 2DE gel which has been stained with Comassi blue. Each spot provides information about the molecular weight and isoelectric point of the proteins.
So, broadly talking about two-dimensional electrophoresis, the very first part you did, the first dimension separation in the isoelectric focusing, when you are looking at there is pH equals to Pi, there is no net charge and proteins are separated based on that depending on what IPG strip you have used. Then in the second dimension, we separated protein further in the SGS page based on their molecular weight property, and proteins are migrating based on their size. So, this kind of technology of two dimensional electrophoresis could be used for studying the differential protein analysis, and that is widely used for many applications. A lot of biological applications have used two dimensional electrophoresis to separate thousands of proteins on the gel, and then they have compared a condition A with condition B. For example, healthy individuals with a diseased individual. So, you first want to separate the proteins from these two populations. You want to solubilize the proteins, you want to separate them on the IPG strips. Then after doing the equilibration step, you want to separate them on the second dimension as shown on the schematic on the screen. Then you are staining them with one of the staining reagents, and after staining, then you can start visualizing thousands of spots on the gel, and then the analysis becomes very crucial. The automation in the software and the our capability for data analysis becomes very critical here, because you want to now pick up the differential protein responses or a few very unique proteins which might have emerged in a disease condition. So, you want to pick up those interesting proteins, and now from the gel you can simply excise those protein spots which are based on the 2D separation. So, once you have excised the proteins of interest, which is the spot you are seeing on the gel, and then we can further process that with some enzymes like trypsin, which convert the protein to peptide forms, and then we can further analyze them using mass spectrometry to identify the proteins. So, today we have tried to learn about some of the basic electrophoretic techniques for the protein separation, and we have mainly studied the workflow of two dimensional electrophoresis, which is one of the interesting, simple, but very elegant technology which can separate thousands of protein. Of course, it becomes little challenging when we talk about clinical samples, because you have large number of samples to be analyzed from the control versus test conditions, and that is where there are some limitation which people start encountering. So, then as a part of 2D electrophoresis, I also tried to brief you about the SGS page gels, which is separating proteins on the molecular weight basis. So, by now you have got good concept of how two dimensional electrophoresis can be performed. Let us start a laboratory demonstration session where my TAs will show you the various steps involved in performing 2D electrophoresis, starting from rehydration, then doing isoelectric focusing, equilibration steps, and performing the second dimension separation. So, let us start the SGS page lab demonstration session now.
Hello, I am Shalini. I am a TA of this course. So, today we are going to learn how to make SGS gel and how to cast it and then how to run it. So, first of all, I would like to explain about SGS. So, that is a technique using which we can separate all the proteins on the basis of its mass. So, these are some buffers which we will require. Like one is 1.5 molar tris buffer solution of 6.8 pH. Other one is tris buffer, and that is 8.8 pH. The other is 10% SGS solution. This is 30% acrylamide and bisacrylamide solution. Here it is timid, and here it is APS. So, all these three are required to be stored at 4 degrees Celsius because these are very sensitive. So, we have already taken aliquots of it and will store it. This is glass slide which is having spacers. This is called thick plate and this is called thin plate, which is separated by these spacers, and here you can see it is mentioned 1.0 mm, which is the So this is a composition in which we APS and timid, and it is to be removed from fridge just before the experiment to be started, before you want to cast the gel. So here we have added everything except timid and APS, so I'll add it. First I am adding APS, that is 50 microliter for one gel, that is. Now I am adding timid, that is 2 microliter. It's electrodes in it. Now we have to place it in such a way that the well are inside, facing inside. So carefully place it here, and now as we are running only one gel, we need a dummy plate which will go on the other side of the gel running unit. Now we have to close it. Now this is sealed. Now we have to transfer this whole unit in the buffer tank, which is having 1x SGS running buffer. As you can see here, there is a red marking and here there is a black marking. So we have to place it in such a way that red comes along the side of red. Now the setup is complete and we have to pour some more buffer inside this running unit. So now this is 1x SGS running buffer. We have to add this in the unit. Now the loading is done. We can remove this, and now we have to start running the gel. So just remember, red color goes with red and black color goes with black. For manual, 90 volt till the time it is getting stacked, and after that when it is stacked we can shift it, we can switch it to 120 volts. So now we will start running the gel. Bubbles can be seen from here, that means the gel has started running. After the gel is run, then we have to stain and destain the gel to see the bands which are separated on the basis of mass using SGS gel. For staining, we have placed it in a plastic plate. As you can see here, the gel is there and we have added Comasi blue, and will keep it for approximately 2 to 3 hours for getting stained. And after staining it, we have to add the destain to it, and after destaining for 2 to 3 hours again, gel looks something like this. As we can see, bands are separated on the basis of their mass. That's all for the SGS page. Thank you.
Hello everyone. Today we will discuss the overall workflow of 2D. 2D and dyes are very important and powerful gel-based proteomic approaches, and both of them, they separate proteins on the basis of isoelectric point and molecular weight. So as you all have studied the overall workflow of one-dimensional gel electrophoresis, the SDS page method, dyes is a step ahead of SDS page. So SDS page separates proteins only on the basis of molecular weight. 2D and dyes, on the other hand, they separate proteins on the basis of both isoelectric point and molecular weight. So one may wonder, what is isoelectric point? So all the proteins, they have a particular pH value at which their net charge is 0. So at isoelectric point of a protein, which may vary from 2 to 7, the overall charge on the protein is 0. So at that point of time, the proteins, they have least mobility. So if we have introduced proteins in an electric field, so when they reach up to a pH value which matches with their isoelectric point, their overall charge will be 0 and they will not move any further, they will just stop there. So that is the principle that we exploit in 2D. So as the name suggests, separation happens in 2 dimensions. The first dimension is known as isoelectric focusing and the second dimension is known as SDS page. So first we will see how isoelectric focusing works. So we have commercially available IPG strips. So IPG strips, they are available in various forms. So in this particular case, this IPG strip, this has a pH range of 4 to 7 and it is 13 centimeter in length. So we have IPG strips which have higher length, or they have different pH range like 7 to 10 or 3 to 10, depending on your sample and what you want to see. So what we will do is, first we will take the IPG strip. Protein sample. On top of that, we will add our IPG strip, and the first step, rehydration, will happen for overnight. So that helps our IPG strip to rehydrate and it will absorb all the protein sample. So this is our protein sample. It has all the necessary components. Our protein is there, urea is there, we have rehydration. Spread uniformly. the negative end. Each IPG strip has a barcode. You can also see there is a pH range mentioned on it, 4 to 7. So what I will do is, I will remove the plastic film, the protective plastic film, and I will keep the gel side. So the gel side is being protected by the plastic film, and I will keep the IPG strip on this tray in a way that the gel is facing the sample. Again, we have placed our strip in a way that the positive end of the IPG strip is facing towards the positive end of the unit and the negative end towards the negative end of the unit. Now we will start the isoelectric focusing. We have also added paper wicks which will soak any salt or all the oils in the sample and help in the proper flow of current. So now we will also add electrodes at both the ends. So electrodes are same for both the ends. And then we will start the isoelectric focusing program, which will go on for 15 to 16 hours. So as you can see, these are the various voltage run parameters. Initially we keep the voltage at 200 volts for 4 hours, which we then increase to 500 volts, further to 1000, and from 1000 there is a gradient increase, and at 8000 volts we keep it for 64,000 volt hours. So this will ensure that a proper IEF run has been achieved. And this is an example where there was some problem in the sample and proper IEF run could not be achieved. You can see this declining red line. It can be because there are any salts or any other components which are interfering. but from IEF onwards we will keep it in a way that the gel side is facing upwards, because proteins have already separated on the gel strip, and we do not want them to get disturbed. So, we will make sure that the gel side is always upwards. So, we will incubate it for 15 minutes. Generally we do it using a shaker, so that the solution A is in constant touch with our IPG strip. So, once incubation with solution A is over, then we will add solution B. Proteins, they have migrated, they have traveled during the IF run, and wherever they found they found a pH value which matches with their isoelectric point, they stopped moving. So, their mobility stopped. So, proteins, you will find it all across the strip. Now, the separation is going to happen vertically. Earlier the separation happened horizontally. Now, the proteins will move according to their molecular weight. So, this is the cassette. We have added our IPG strip. We have also added some agarose solution to seal it, so that the strip does not move here and there and it is static. So, we will then just assemble it. The way we do for normal STS page gels, we will then destain, and then this image is one of the representative images which you will see after you have destained your gel. You can see there are multiple spots on this image, and then later you have to annotate these spots using various softwares and identify your protein of interest.
So, I hope you had a good view of processes involved in performing 2D electrophoresis. You have seen there are multiple steps involved, and which makes the process a little tedious, but if you have done everything meticulously, then probably you can see some beautiful spots which are separated on the gel, and those gel images reflects your hard work, your efforts which have been putting to know the protocol, the science, all the steps involved in doing good experiment. And each spot actually reflects what is the isoelectric point, what is the molecular weight of a given protein. So, I hope now you have a much confident understanding of performing 2D gel based proteomics experiment. In the continuing lecture, we are going to discuss about some of the pros and cons of using 2D electrophoresis and how it can be made more robust and more quantitative for various other applications. So, let us continue the discussion on technologies in the next lecture.